Next up, we have Raymond Cloyd, who is going to be discussing how to implement a successful biological program. So Raymond Cloyd is a professor and extension specialist in horticultural entomology and plant protection in the Department of Entomology at Kansas State University. His research and extension program involved vegetables and fruits, and plant protection in greenhouses, nurseries, landscapes, and conservatories. Raymond, we're glad to have you and look forward to hearing your presentation.
Hear me? So.
Yep, we can hear you.
Can you hear me clearly?
So far, so good.
Okay, so I'm going to go ahead, and if there's any problems, is start my presentation. Is it up there? Hello?
Yep, we can see it.
Okay, can I close the window on the right, Karen, or do I need to leave it open?
No, we can't see it. The only thing we can see is your PowerPoint, so you're all set there.
Okay, hold on. Let me just. Got it. Okay, so good morning or afternoon, depending on where you're located. I'm going to be talking about how to implement a successful biological control program, what you can expect. It's not advancing. Why is it not advancing?
Click on your screen, and that should work.
Okay, thank you. Thank you, Karen. What we'll talk about during my the first full presentation is give you an overview of what to expect. We'll give you a brief introduction, talk about the basics of biological control, talk about the biocontrol agents, also referred to natural enemies, and then how to implement a successful biological control program, and then at the very end, questions and discussion. Obviously, this is an example of greenhouse production horticultural crops. However, when you're growing horticultural crops, you're more likely to run into a series of insect or mite pests. That includes aphids, fungus ant larvae, or the two-spotted spider mite. In general, the primary means of dealing with insect or mite pests in greenhouse production systems has been the use of pesticides, insecticides, and miticides, and that can include conventional and some of the biopesticides commercially available, which have been discussed by some of the sponsors. However, there is an alternative plant protection pest management strategy called biological control. It's been around for a long time, since the 50s and 60s. And so this is just an example of what happens when biocontrol products are commercially available from various distributors and suppliers throughout the United States. However, before we move on, I need to talk about why growers are interested in implementing biocontrol programs. Basically, it's threefold. There are fewer new active ingredients being introduced because of rules and regulations and the cost of discovery and then also marketing. Number two is to avoid the issues associated with insecticide and miticide resistance. There's a number of materials, I think Conserve stands out that when it first came out was highly effective on western flower thrips, but because of continual use has lost its efficacy. And also the issues associated with neonicotinoid systemic insecticides in bees. Number three is the interest by the public for plants produced organically or more so minimal inputs from pesticides. And this is mostly associated with the residues on plant parts. So these are just some of the reasons that I've gathered through growers talking about why they want to implement a biocontrol program. So what is biological control? Biological control is the release of natural enemies, in this case parasitoids and predators, to regulate, and the key word is regulate, insect or mite pest populations. Biological control agents will not eradicate pest populations. So the success of a biocontrol program is contingent on maintaining insect or mite pest numbers at levels low enough to minimize plant damage. So what I tell growers is 2 weeks before you ship, look at your crop. If you don't notice any major damage or anything like that, you've done your job. And that's what the biological controls are designed to do, is to regulate insect or mite pest populations and keep damage to a minimal. So there are 2 approaches to biological control. The first one is we call augmentation. That's where you basically increase the number of existing natural enemies. And there are 2 approaches. One of them is inundation. The second one is inoculation. The other one is called conservation biocontrol, and this is not used in greenhouse production systems. It's mostly used outdoors in nurseries and vegetable crop systems where you put in plants that protect and maintain existing populations of natural enemies. So let's go ahead and delve more into the augmentative approach of biological control. Well, augmentation is the practice of purchasing natural enemies from a supplier distributor, and then you release them out into the greenhouse system. Now, inundation is releasing biocontrol agents in order to quickly suppress insect or mite pest populations, and you're not relying on the subsequent progeny or next generation to provide any level of suppression. This is using biologicals similar to using an insecticide. The second approach is inoculation. And that's where you're releasing small numbers of biocontrol agents over a prolonged period. And you're not, and this is where you are expecting the progeny or next generation to provide some level of regulation or suppression. Inoculation is most widely used in longer-term cropping systems. So here's an example of an operation near my office where they're using these sachets and they're releasing predatory mites, and you can see every one of the flats has a sachet containing predatory mites. And this would be an example of inundative use of the biocontrol agents. So you're going to start a biocontrol program. What are the requirements? Well, you have to know the availability of biocontrol agents or natural enemies by looking at some of the catalogs of the biological control producers and suppliers. Number two, you have to understand the biology, life cycle, and the behavior of the insect or mite pests within the cropping systems. Number three is scouting crops regularly, and I will dwell more on this on my lightning round, and ordering biocontrol agents as needed. Remember, biological control is a preventative process. It is not reactive. You cannot release natural enemies when you've got 5 million aphids on your plants. Heaven forbid you have that. But you have to release them almost, and I'll say almost, before you even see the pest on the crop. And then the last one is you have to conduct follow-up to assess the failure or successes of the program. You will run into stumbling blocks, and that's expected. But the more you try biocontrol and get used to it, the more you're likely to succeed. So I want to highlight an article by Albert Grimm. He's a grower in St. Catharines, Ontario, Canada, works for Jeffrey's Greenhouse, and he wrote a really good article that was published in Grower Talks regarding the biological black holes. And in his article, he talks about, this is a paragraph that I recommend you read, talking about how our system, our approaches to biological control are very different from the use of insecticides and miticides, and what we should expect from biocontrol agents when they're being released in greenhouse production systems. So again, don't have time to read the entire article, but I do suggest you download it and give it a good read because it is highly educational, and one of the most important aspects of it is it's very practical. The other aspect of biocontrol and why it's preventative is that the reproductive capacity of certain insect and mite pests, in particular aphid on the left and two-spotted spider mites, they reproduce so many offspring that the biocontrol agents cannot effectively regulate them to numbers that would minimize any plant damage. So again, this highlights the relevance and the importance of preventative releases of biologicals and not reactive. So there is a wide variety of biocontrol agents commercially available from many different producers and distributors, which I will highlight at the very end of this presentation. But I want to talk about the types of biocontrol agents just to make sure that everybody is on the same level. There are three types: parasitoids, predators, and then also beneficial nematodes. So this image, here's a parasitoid on the left top. That's a Incarcer famosa greenhouse whitefly parasitoid. And on the right top is Neocoelus cucumaris. That's a predatory mite of western flower thrips. On the bottom left are beneficial nematodes. And then also another group is called the beneficial fungi. And these include products like Botanigard, Ancora, and Met52. They're biopesticides bottled, and then you spray them on plants. So some of us don't put them in the same category as biological control agents or true natural enemies. So what are the categories of biological control agents? Well, the first one is specialist. Specialists feed only on one insect or mite prey or a particular life stage. It could be the egg, larva, nymph, or adult. These are generally parasitoids. Mostly parasitoids, we call them specialists because they only attack one type of insect or mite and they only attack one particular life stage. The second category are the generalists, and they feed on a wide variety of insect or mite hosts, and they tend to feed on more than one life stage. And in particular, some predators, and these are mostly predators, will be predaceous as larvae and adults. That's not always the case, and I will make that point out. But in some cases, you'll get the larvae and the adults are going to prey on different food sources and also different life stages of particular prey. So let's talk about parasitoids. Aphidus aerivii at the top, Aphidus kalmanii at the bottom, those are aphid parasitoids. Over here on the top right is Encarcer formosa, and the bottom is what happens to a whitefly that's been parasitized by Encarcer formosa. It turns distinctly black. If it was not parasitized, it would be basically translucent, which is shown on the top part of that image. So let me go over how this works. The female parasitoid takes her abdomen and uses her ovipositor, which is the egg-laying device, into this unsuspecting aphid. Now that egg will hatch into a larva that eats the inside or the internal contents of the aphid, and at the same time, I have to say this, gives off gases, or in layman's term, farts. And what that does then is causes expansion of the aphid, and you get these gray or brown encased mummies, mummified aphids or parasitized aphids. So this is an example of these are aphids that have been parasitized by a parasitoid, or we also call them parasitic wasps. So if you're still having trouble understanding this, if you've seen these two movies, then you know what a parasitoid is. If you remember in Alien, when John Hurt in the dinner table had something come out of his chest, that would be a good example of a parasitoid. In addition to direct laying of eggs in the host prey, some parasitoid females will actually host feed. And here's Eretmosferus aurymicus. It's a whitefly parasitoid, and is using an antenna to determine the quality of the prey, and it also will use it by feeding on it. And some of this host feeding can result in some level of mortality of the whitefly nymph. Now, what are the ways to release parasitoids? Well, you can get these containers, which are filled with mummies, and you place them among the crop, but mostly these are pupa that are attached to cards that you basically put on plants. And here's an example of one of the cards. In the center, in the circle here, are parasitized pupa, and within 1 or 2 days, the adults should come out of there and start looking for whitefly nymphs. You want to be sure to don't put these in direct sunlight and don't get them wet, as they will rot. Okay, we'll talk about monitoring. When you're using yellow sticky cards, well, you got to take them down because parasitoid adults are attracted to that wavelength of the yellow and can be captured. So you don't want to release parasitoids. Come to find out, all of them have been captured on the yellow sticky cards. Not an efficient use of biological control. So let's talk about the aphid parasitoids. Well, this is why it's important to identify the aphid species, because not all parasitoids attack all aphid species. For example, Aphidus aerivii parasitizes what we call large aphid species, which includes potato and foxglove aphid. Whereas Aphidus kalmanii parasitizes a small aphid species, and that includes green peach and cotton and melon aphid. Now, Aphelinus abdomalis parasitizes potato and foxglove aphid and other aphid species. And then Aphidus matricaria parasitizes green peach aphid. So again, it's really critical if you're going to use biological control with parasitoids for aphids that you get them identified to species. Now, however, if you're dealing with a multiple complex of aphids, then you can get a combination of Aphidus kalmanii and aerivii. That way you get green peach and cotton and melon and potato and foxglove aphid. This typically occurs when you're growing a multitude of crops which are susceptible to different aphid species. You have to understand that environmental conditions, primarily temperature, will influence all natural enemies. For example, Aphidas airvi, the life cycle takes 14 days at 7 degrees Fahrenheit and 20 degrees at 59 degrees Fahrenheit. The higher the temperature, the shorter the life cycle will be because all insects and mites are cold-blooded. Aphidas colmani, the optimum temperature for development and parasitism is around 86 degrees Fahrenheit. So again, it's going to be critically important that not just releasing these, but understanding their optimal temperatures that will help you succeed in implementing a biological control program. Now, here's a publication. I read lots of papers, and they're always chock full of knowledge. But in this one, they talk about the optimal temperatures for development of A. colmani and Metricaria were around 30 to 25 degrees C, and higher mortality occurred at temperatures that were greater than that. So again, you have to understand the optimal temperatures that are going to be desirable for these natural enemies to succeed. Now let's switch to predators. We have Neoseius californicus, Neoseius cucumaris, and the workhorse is Phytoseius persimilis. Phytoseius persimilis is a predatory mite for two-spotted spider mite. Well, this is some of the formulations and releases. You can buy them in containers, but more growers are using them where they hang them on the plants, or they're using what we call the popsicle sticks. You insert them into the containers, and the mites will come out of these sachets and they get onto the plant looking for different types of prey. Now here's a container with about 250,000 predatory mites, and I really recommend that you make sure you're getting your money's worth by counting all of them, and so you're not getting ripped off. Obviously, I'm kidding, but this is a large size of Amblyseius cucumaris that you can distribute by hand, or I'll show you another means within the greenhouse production system. Here is a product containing 25,000 Stratolalia semitis, which used to be called Hypoacis miles. This is a predatory mite for fungus gnat larvae and western flower thrift's pupa. Here is the old method, we call it the salt and pepper shaker method of releasing these, where you have a bran or vermiculite, you shake them over the plants, and the predatory mites then distribute themselves above, around the plant canopy, or if you're applying the soil-dwelling predatory mites, they'll start moving into the growing medium. Here's an example of a grower that's putting Phytoseius persimilis among the crop, and look at the size of the crop. Those are seedlings, but again, biocontrol is a preventative process. So you're going to release the biological control agents long before you even see possibly the pest on the crop. Here's another greenhouse operation where they're using, this is all herb production, and they're using these sachets to release predatory mites. So when you get close up, you can see that this is called inundative use, where they're putting a lot of these sachets out to basically overwhelm the potential pests that might be attacking these various herb crops. Here's a system where they're using these predatory mites for hanging baskets. And one of the systems that growers are using is they're applying mainspring, which is an insecticide to the soil as a growing medium, and that will control aphids and the larval and adult stages of thrips feeding on the foliage, but they also add the mites to clean up any thrips that escape the exposure. So this is being used widely for hanging baskets, because once pests get into hanging baskets, growers do not want to start bringing them down and spraying them. So hanging baskets are very conducive for the use of biological controls, in this case, predatory mites. So instead of using the salt and pepper shaker method, there's been some mechanical means of releasing biocontrol agents. And here is, on the left, is a blower, battery powered blower, where they have cut a hole, and now they have a system of metering out the mites and spreading them out throughout the greenhouse. Here's a more recent one I saw where they're using the Venturi effect, and as this blower is going out, the mites get picked up with the sawdust and are blown throughout the greenhouse. Studies conducted at Kansas State and other universities have shown these to be very, very effective in releasing predatory mites. Now, I don't expect you to read and understand all this. My point with this image is that there is a lot of different predatory mites. Phytoseius persimilis is a predatory mite only for two-spotted spider mite, and it requires certain relative humidity and temperature conditions. Many of these other predatory mites that are available will tolerate higher relative humidities or lower temperatures than Phytoceus persimilis. But I just wanted to give you a listing of these predatory mites and the target insect or mite pests that they're designed to regulate. So there are 3 types of predatory mites. They're type 1. These are called specialist predatory mites. And this includes Phytoceus persimilis. It only feeds on 2-spotted spider mite. So once the food quality or food is gone, they will either probably die out. There are type 2 predatory mites, we're called selective, and these feed on a broad range of prey and will also feed on pollen as an alternate food source. Examples include Neoseius californicus and Neoseius cucumaris. The type 3 generalist predatory mites feed on a very broad range of prey, including areophyte mites, that includes the tomato russet mite, and the broad encyclone mite. This includes Amblyseius swirsky and Californicus. Now, as you notice, Californicus is both a type 2 and a type 3 predatory mite. So again, going back to the environmental conditions and how they impact beneficial insects or biological control agents, here is a study looking at the impact of temperature and relative humidity on Phytoceus persimilis. Phytoceus persimilis requires a relative humidity around near 70–80% and temperatures between about 65 and 80 degrees Fahrenheit. What happens if you get outside those ranges, you have problems like lower egg hatches, and you get fewer nymphs and adults on the plants. So that's why you have to understand the optimal temperature conditions for the biological control agents you're going to release. And again, here's a publication, and it says the results showed that more prey are eaten at as temperature increases from 15 degrees C to 25 degrees C. But once you get above that, the feeding ability of Phytoceus persimilis starts to decline. You know, in the case that you're kind of concerned that you're worried there's not enough prey available, then you can release supplemental food sources such as Nutrimite, which contains cattail pollen. Cattail pollen has been shown to be a food source for certain predatory mites, including Amblyseius swirsky, Neoseius cucumaris, and Neoseius californicus. So early in the production cycle, you can actually use a supplemental food source to retain populations of these types of predatory mites. Well, in addition to predatory mites, we have other predators, including insidious flower bug and row beetles. This is the insidious flower bug. There is how small they are compared to a dime. They are predators of primarily western flower thrips. Both the nymphs and the adults are predaceous. Then we have row beetles. This is Delosia coriara, and the top is the adult row beetle. And although hard to see, I've highlighted, this is the larval stage. These are soil-dwelling predators, and they feed on fungus ant larvae, shorefly larvae, and our current studies have shown they will also feed on western flower thrips pupal stages. Both the adults and the larvae are predaceous. The other predator are green lacewings. Here's a green lacewing right there on a card. These are the eggs, which are on stalks, and they feed primarily on aphids, as you can see in this image on the upper left corner. And here is how they're released. This is an operation that grows hydroponic lettuce, and these are all the egg stages. So when the eggs hatch, they crawl out and will start looking for aphids among the lettuce crop. The last group are the beneficial nematodes, or also called entomopathogenic nematodes. And those of you having spaghetti for lunch, you might want to remember this image. They're microscopic roundworms. They need moisture to move around. And they're primarily used as drenches for fungus gnat larvae. And they have been used as spray applications for regulation of western flower thrips on the foliage of certain plant types. But let me explain how this works. If you drench them, they enter the fungus gnat larva through natural openings that includes the mouth, the anus, or the spiracle, which are the breathing pores, and then they release the bacteria. It's the bacteria that actually consumes the internal contents of the insect, and that's what the nematodes feed upon. Then, after mating in several generations, they create so many individuals that they burst out of the dead cadaver. And so here's an example of what happens when you get millions and millions of these nematodes developing, and they break out, and they will go in search of other prey. Now, if they can't find a fungus gnat larva, they will probably die out, which means you have to make repeat applications. And here's just an example of one of the products that contains 50 million nematodes, and you want to be sure you count those so you make sure you're getting your money's worth. That's used for primarily fungus gnat larvae. So, how to succeed using biocontrol? I've talked about the agents. I've talked about introduction. First of all, you have to establish a reliable scouting program. I'll dwell more on this on my lightning round. You have to correctly identify the primary insect or mite pest. You have to identify available biocontrol agents for specific insect and mite pests. You want to establish a relationship with a supplier or distributor of biocontrol agents. You want to minimize pesticide residues. You don't, especially the pyrethroids and those that are very harmful directly to natural enemies. You want to have a clean greenhouse that is no weeds or debris within the greenhouse. And you want to order your biocontrol agents early. Again, it's preventative. I have growers that call the supplier and tell them, send me biocontrol agents every 2 weeks, so I know they're going to come. You also want to make sure the biocontrol agents are alive. We know for studies after studies that dead insects will not regulate live insects, and we'll talk more about that. You want to release your biocontrol agents immediately upon arrival. Many of them are shipped without a food source, and you don't want them to just die of starvation. You want to apply them in the early morning or late evening, give them time to acclimate into their new environment. And you want to introduce them before the insect or mite pests are high or reach out big proportions. Once that occurs, it's too late to implement a biocontrol program. And then you want to evaluate the performance of the biocontrol agents, looking at the failures and successes throughout the growing season. Again, the first item that needs to be considered, you have to have a scouting program, a well-established, reliable scouting program. And again, I'll focus more on this on my lightning round, but this includes using yellow sticky cards or visually inspecting plants. So here's one of our growers. He's identifying the insect on the yellow sticky cards, and then he's recording the data on his data sheets. And this is what he's recording, and I'll talk more about this during the lightning round. But I want to make sure that you understand, if you're going to implement a biocontrol program, you have to have an sound established scouting program. So what are some of the factors that can influence the success of a biological control program? Well, quality control, I've already mentioned the environment, and also the plant characteristics. The one I want to focus in on is, how can I make sure that my biological control agents are alive upon receipt? That's what we're going to cover. So what are some of the quality control issues associated with biocontrol agents, or how can you conduct a quality control program? First of all, when you receive biocontrol agents, make sure they're packed adequately. Make sure they're in styrofoam containers, peanuts or newspaper, and generally most of them will send you a device which has monitored the temperature from when they send them to you and then when you get them. Because a lot of times during the shipping process, that's where we find most problems associated with mortality of biocontrol agents. You need to make sure, are they alive when you receive them? Here's a container with styrofoam, and you should also have peanuts or newspaper so then they don't get jostled during the shipping process. Again, when you get your biocontrols, are they alive and are they functional? For example, you get a box of these predatory mites and sachets. How do you know that all the sachets contain the same number of predatory mites? So here we're going to talk about quality control for predatory mites. This is in our laboratory, and we're getting ready to take the popsicle sticks and determine if there's live or dead predatory mites in them. So what we do is we break them up and we look for anything moving around. Now remember, these are typically shipped with grain or bran mites to keep them alive, so you don't want to count those as predatory mites. Well, if this is too laborious and you're worried about, I can't tell the difference between a good mite or a bran mite, then you can do this. You can take a yellow sticky card, you can take the popsicle sticks, bend it. Now, when the predatory mites come out, they'll crawl down and get stuck on the yellow sticky card, or they'll come down and drop down. And there you can tell, you won't want to count all of them, if the predatory mites are emerging from these sachets. Now, what about parasitoids? Well, what you can do, you can take the cards, put them into a mason jar, and then you're going to want to attach a section of a yellow sticky card to the bottom. Wait about a week, take the lid off, and then you can tell the number of adult parasitoids that have been captured on the yellow sticky card. Here's another example that we've done in our lab where we put one of the cards, which is supposed to be whitefly parasitoids, and under the lid, we put a yellow sticky card. After about 5 days to a week, we take the lid off and then we look at the number of parasitoids that have been captured on the yellow sticky card. That gives us an idea of the emergence of functional individuals. What about nematodes? Well, live nematodes should be curly and moving around. Dead nematodes are straight. Well, you can just take a glass beaker or vial, take a section of this formulation of the nematodes, fill it with half full of water, hold it up to the light, and you should see the nematodes moving around. If they're not, you want to call the supplier distributor immediately. For example, here's a study one of my graduate students did evaluating the percent live entomopathogenic nematodes for various suppliers. And you can see that one of the suppliers, only over half of them are live and nearly half of them are dead, where the product Nemesis, we were getting 98% live EPNs, which is very, very good. So if you want to read more, here is the publication and the journal where it came into if you want to learn more about quality assessment of beneficial nematodes. So when you get nematodes and you can't release them right away, at least a day, you got to store them properly. And here's an operation we have dealt with in the Midwest that uses lots of biocontrol agents. They store them in a refrigerator and they release them the next day. What's also important to understand is what life stages of a pest are susceptible to natural enemies. Well, let's take Western flower thrips as an example. The Western flower thrips life cycle consists of an egg inserted into the leaves, 2 larval stages, the first instar larva, then 2 pupal stages in the soil. This is the prepupa and the pupa and the adult. Well, let's look at the biocontrol agents and which life stage they attack. Well, Neosis cucumaris only attacks the first instar larva. Amblyosus swirsky attacks the first and maybe the second instar larva. Oris insidiosus, the minute pirate bug, attacks the first, second instar larva and the adult. And then Stratolaelis semitis, formerly Hypoacis miles, attacks the pupal stages. And the roe beetle Delocia coriara also feeds on the pupal stages. So you can now understand that in some cases you have to use more than one biocontrol agent to deal, in this case, with Western flower thrips. So the last thing I'm going to talk about is the banker plant system. What that involves is growing rye or cereal plants that you put in your greenhouse that have an aphid that does not attack the plants in the greenhouse, but serves as a refuge or reservoir for the parasitoids, in this case, aphid parasitoids you're going to release. So this is a banker plant that contains both live and mummified aphids on it. So this is what you want to see on the banker plants. These are mummified or parasitized aphids. So what you do is you can purchase banker plants from one of the suppliers or distributors of biocontrol agents. And then this is where it gets tricky. You buy the pest or something like the bird cherry oat aphid and you apply them to the banker plants. But you have to protect them from natural parasitization, and this is an operation that built a cage, and these are all the banker plants. They're letting the bird cherry oat aphids established before they put them out into the greenhouse. And here's another greenhouse I've worked with where they use cloth to protect the banker plants and the aphids on them from natural parasitism. Another banker plant system involves black purple, black pearl, or purple flash pepper plants, and this is for Aureus, minute pirate bug, and Western flower thrips. So what about dealing with multiple pest complexes? Can biocontrol work in these instances? In general, I recommend targeting the primary insect pest with your biologicals. But if you have thrips and spider mites, then you might have to look at using multiple biological control agents. And this is done on a case-by-case basis. So what about some information out there? Well, I have worked with colleagues in Michigan, and we've developed a fact sheet called Commercially Available Biocontrol Agents for Common Greenhouse Insect Pests. And in there, we have sheets for all the biocontrol agents for the various insect or mite pests. And this is an example for aphids where we have the parasitoids and the predators listed with key biological information. My book is still available, was published in 2016, and it contains an entire chapter on how to use biological control in the greenhouse production systems. Heidi Lindberg and I developed an online course on biological control course for greenhouse growers. There's the website. It consists of 5 units or modules, and it generally comes out, you can register twice per year, but there's the website if you want some more information and if you want to take the course. These are some suppliers of biocontrol agents that I have been involved with that I respect very much. That you can get a lot of good information. They provide good technical support, and they want you to succeed using biological control. So with that, I'm done. And if there's time for any questions, I do thank you for attention. I'd be happy to address those.
Thanks a lot, Raymond. We certainly do have some questions for you.
Excellent.
So we have one here. Why should biologicals be applied in the morning or evening? Is it more due to temperature or light conditions?
Excellent question. Well, when you get these biologicals, they've gone through some travels. They probably have a headache and things like that. But what I was getting across is you don't want to put them on the heat of the day because that may stress them. So it's better to put them out early in the morning, late evening, let them get acclimated, but don't put them out in the heat of the day.
Okay, and you'll have to excuse my very bad pronunciation of these, but I hope you can understand all the ones I'm trying to say. So regarding the yellow sticky tape, we use the strips of yellow hopper tape on the top of the plants where the whitefly usually accumulates at the growing tips to catch the adults. So if I release Encarsia or Iridomyceras, I should not use the rows of yellow hopper tape above them?
That's a great question. If you're using Encarsia or Iridomyceras and you're using the hopper tape or the big tape, yes, if you do release those, they will get captured on that yellow sticky tape, yes.
So you had mentioned earlier in the presentation that the predatory mite needs enough food to be able to survive. So the question here is, if there is no prey or food, how long do you expect the predatory mite to survive?
That's another good question. First of all, it depends on the predatory mite, but let's just take in general, if Phytoceus persimilis cannot find any prey, then and sometimes it'll cannibalize it'll feed on others. It's probably going to take less than a week for it to finally starve to death. Yes.
Okay, we have a couple people asking about this one. Are there any biological controls for mealybug?
Excellent question. One of my pet peeves is, yeah, there are biocontrol agents for mealybugs. The first one is the predator, Cryptolemus montrosieri, which is called the mealybug destroyer. However, that predator, it's a ladybird beetle, requires a high numbers of mealybugs that you really couldn't accept in a greenhouse production system. There are some parasitoids. Unfortunately, they're limited availability. One of them is Leptomastix dactylopi. I don't know if that's available anymore. And the other one is Anagyrus pseudococcidae. And I do believe there's a company in Israel that has the rights to ship that to the United States. So from a biological standpoint, mealybugs are very limited. We actually need more biologicals for mealybugs. Okay,
so this one, we have a question regarding banker plants. So with banker plants for cherry aphids, usually in spring and winter, it is cool and barley seems to work well. With the hotter weather in the summer, barley sometimes does not survive well. What would you recommend instead? We try to put the banker plants as hanging pots above the cut flower crop.
Yeah, that's a good question. I know others have used wheat or rye, and I think they do a little bit better than barley in the spring and summer. Yeah, I would try those.
Okay, and then a question about compatibility. Are all biological agents compatible with each other?
Um, I'm that's a great question, and it really would be a webinar by itself. But yeah, we do know that some of the biologicals out there are not compatible. You can't use them together because one of them will eat the other one. So that's a great question, but it really requires another webinar or more time to really address it. But yes, the general consensus is that not all biologicals can be used together because some, especially predatory mites, they will eat each other if they can't find a food source, and so that means they would not be. You couldn't use them together or in conjunction.
Okay, one last question here. Are there any good biologicals for root aphids?
Yeah, the question of the year. The answer definitively is no, but the calls that I've been getting from certain growers, and we won't talk about that, is that I have been recommending row beetles Delocia. I got no data, but Delocia is a general predator in the soil, and if there's going to be any biological that might work on root aphids, I would think that would be the one to try.
Great. Well, I'll definitely be sending you the remaining questions that we couldn't get to now, Raymond. But we
Okay,
look
Karen.
forward to hearing from you again in about 10 minutes.
I'll be ready.